So in this video I'm going to explain you about the biofilm formation. So biomaterial associated infection always leads to biofilm formation. So biofilm is a complex community of microorganisms that adheres to the surface of a biomaterial device and are embedded in self-produced EPS matrix. Here EPS represents extracellular polymeric substance. This prevents the biofilm from the removal by the environmental stresses. So actually biofilm constitutes the protective mode for the bacteria to reside in host tissue, preventing it from the removal forces, allows nutrient uptake and waste removal process, and it also further protects from the host natural responses and antibiotics. So it makes the microorganisms antibiotic resistance and also resistant from the host natural immune defenses. So steps in biofilm formation. So the most important virulent factor for bacterial associated biomaterial infection is bacterial adherence. So for that, bacteria adheres on the surface of a biomaterial by weak interactions like covalent, polar or hydrogen bond interactions. This is initial attachment and this can be reversible. It means in this case they can be detached to irreversible attachment. Now if the bacteria adheres on the surface by pili, fimbriae or EPS, where EPS is extracellular polymeric substance matrix. So this makes the attachment irreversible. Now this EPS prevents from the removal by the environmental stresses. Now growth and development. Adhered cell starts to proliferate and biofilm grows into a structured and multi-layered community. Then after that maturation occurs. The biofilm develops into a complex 3D structure with waste channels for waste removal and also allows optimization of effective nutrients trace elements to the final stage is dispersion. Here the biofilm leads to the dispersion. Now some of the cells detach and disperse to colonize new surfaces. So I have drawn here the picture too. These are the freely living planktonic bacteria. Bacteria. So first step is adherence. So they adhere on a surface of a biomaterial. Surface of a biomaterial. Then they will grow and multiply and form a multi-layer. Now after maturation, the biofilm develops into a complex 3D structure. This is the 3D complex structure, and now there is dispersion. It means these grown and mature bacteria again complete this cycle on the other places. So they even lead to the secondary cycle of biofilm formation.